Hi, everyone. This is Richa Singhania, Director of the Brain Organoid Core at Weill Cornell Medicine, New York. Today, I'll be talking about new organoid-based models of glioblastoma and how they're helping us unlock the complexities of this terrible cancer. I want to first talk about glioblastoma multiforme, or GBM, which is the most frequent and deadliest primary brain tumor in adults. GBMs are classified as grade 4 gliomas by WHO as they are highly proliferative, infiltrative, necrotic, neoantigenic, and are associated with an immunosuppressive environment. GBM treatment mainly consists of a surgery followed by adjuvant radiotherapy and chemotherapy based on an alkylating agent called temozolomide. The standard of care treatment has remained unchanged since 2005, and no major clinical advances have been achieved to increase patients' overall survival. As you can see here, the median survival of these patients is a mere 15 months after initial diagnosis, and fewer than 6% of patients survive for 5 years after diagnosis. Tumor reoccurrence is seen in 90% of the cases, because complete surgical resection is almost never possible, and there are currently no therapies to eradicate what are called as glioblastoma stem cells, or GSCs, that possess tumor-initiating potential and exhibit resistance to therapies. Despite extensive research efforts, treating GBM still remains a therapeutic challenge. This is primarily because there are three prominent features that make these tumors deadly. First, they're highly invasive and aggressively infiltrate the brain, as you can see in this picture. Therefore, complete resection is almost never possible. Second, tumor resistance. GBM tumors have limited response to chemo and radiation therapies because we now know that these tumors are supported by networks called as tumor microtubes, which allow these tumor cells to communicate with each other and protect them from cytotoxicity. And third, these tumors display a substantial genetic, epigenetic, and transcriptional heterogeneity, both within the same tumor and between the tumors. Additionally, recent single-cell sequencing studies have provided evidence for something called as GBM subtype plasticity, meaning that multiple subtypes can coexist in the same tumor, and these subtypes can change over time and through therapy, which adds another layer to GBM heterogeneity, making it extremely difficult to treat. Here I'll discuss some of the most widely used in vitro and in vivo models of GBM. Isolation of primary patient-derived glioma stem cells from fresh tumor tissue and culturing them as free-floating spheres was a significant advance from using immortalized glioma cell lines, which were rarely of any clinical significance. The tumosphere model relies on the capability of tumor cells with stem-like properties to self-renew and offers the advantage to be representative of the genetics of the parental patient tumors. However, sometimes sorting for a specific cell population before sphere culture could add bias. The main limitation of this model is the lack of cells of the GBM microenvironment, which prevents the study of interactions between GSCs and their neighboring host cells. But they do offer the advantage to be used in high-throughput screening assays as they go fast and are easy to manipulate. And since they are derived from patients, they can also be used in personalized assays. Furthermore, they are used to derive other GBM models, which I'll discuss in a moment, such as tumor organoids. So tumor organoids aim to reconstitute a tumor in a dish for long-term culture by basically culturing glioma stem cells within the extracellular matrix on a shaker. They resemble simple spheroids, which are made of cancer stem cells only, and as a result, they lack the organ-like histology and the cells of the local tumor microenvironment, just like the GSC tumospheres. To reconstitute local microenvironment, organotypic slice culture has been adapted to graft GSCs on a slice of healthy mouse brain. However, these slices can only be maintained for a few weeks, and slices from different areas of the brain would have to be needed to precisely evaluate the impact of entire brain microenvironment on the tumor growth. And moreover, for this method, rodent brain is used, which fails to obviously capture the complexities of the human brain. Lastly, patient-derived xenograft models, where primary GSCs are transplanted in a rodent brain, or genetically engineered mouse models, are powerful in vivo models to study GBM pathophysiology, but they poorly capture the invasive behavior of the human tumors, which I just showed you in the previous slide. So the tumor grows more like a solid tumor mass, which is not really what happens in a human patient. And as everyone knows, mouse models are time-intensive, technically challenging, plagued by cross-species tumor-host interactions, and are obviously not suitable for high-throughput screening. A wealth of information has been gathered from all these model systems. However, translation of these findings has not seen much success in the clinic, partly because all of these models are limited by the lack of human-specific tumor microenvironment, which is extremely critical for modeling human GBM. Therefore, new models are urgently needed that accurately represent these complex characteristics of these tumors, such as the invasive behavior of GSCs, extreme tumor heterogeneity, crosstalk of these tumor cells with their microenvironment, their resistance to the therapies, and problem of immune and the immune escape that these tumors evade the human immune response. A breakthrough in neuroscience research was made when Lancaster from Jürgen Noblich's lab in Vienna published these seminal papers on generating cerebral organoids, or they're also called as whole brain organoids. These are three-dimensional self-organizing brain tissues that mimic developing human brain and offer a fantastic model system for studying human brain development and neurological conditions, including brain cancer. So before I delve into how organoids can be used to grow GBM tumor in a dish, let me spend a couple of slides to give you some background on what cerebral organoids are. So cerebral organoid production begins with generating spheroid embryoid bodies from human pluripotent stem cells, such as embryonic stem cells or induced pluripotent stem cells in ultra-low attachment plates. EVs are then cultured in the absence of serum and growth factors that promote spontaneous induction of a polarized neuroectoderm, which is characterized by a smooth and translucent layer on the outer surface of EBs, as you can see here. EBs are then cultured in neural induction media, which promotes neuroectodermal layer to undergo expansion to form neural rosettes. For further differentiation, these 3D structures are embedded within matrigel, and the ECM provided by the matrigel promotes the outgrowth of large neuroepithelial buds, which then spontaneously expand and develop into various brain regions in the absence of exogenous patterning factors. Thereafter, these organoids are cultured in maturation media on an orbital shaker to grow and develop into mature organoids containing brain lobes with fluid-filled cavities that resemble brain ventricles. This entire process takes about 4 to 6 weeks, after which the organoids can be maintained under shaking culture conditions to mature over many months. Individual organoids can reach up to few millimeters in size upon maturation. So cerebral organoids can model the developing human brain with remarkable fidelity and recapitulate many aspects of embryonic cortical development. H&E sections of our cerebral organoids show similarity to a fetal brain at 20 weeks of gestation. Furthermore, molecular analyses conducted in these papers to compare organoids and fetal neocortex have demonstrated that cerebral organoids recapitulate transcriptomic features of fetal brain development and show that these organoids use genetic programs very similar to fetal tissue to generate a structured cerebral cortex. Organoids recapitulate structural and organizational features of the developing human brain. Younger organoids display a primitive ventricular system with multiple neural tube-like structures called as neural rosettes. And individual lobes show the presence of a radial ventricular zone-like structure with a proliferative zone of neural stem cells and migrating neuroblasts. Mature organoids display brain microanatomy with a variety of brain regions, including predominantly the forebrain region, limited midbrain and hidden-brain identity, hippocampus region, as well as choroid plexus epithelium and retinal tissue. They also exhibit cortical plate layering that is reminiscent of a developing cortex. Organoids also capture cellular composition of the developing human brain and contain diversity of cell types at different stages of development. Younger organoids show the presence of neural progenitors as evident by Nestin staining. These neural rosettes stain positive for ventricular zone marker PAC6 and subventricular marker TBR2. Lumens of these rosettes are positive for the tight junction protein N-cadherin. And mature organoids show the presence of neurons evident by positive staining for MAP2 and TUJ1, as well as they have GFAP positive astrocytes. Presence of cortical plate neurons is marked by CTIP2, and positive TTR and RPE65 staining confirms differentiated choroid plexus and retinal development, respectively. So now I want to talk about how we have applied the brain organoid platform towards modeling the pathophysiology of human GBM and applying diverse therapeutic interventions. We've developed a robust 3D model to study human gliomas, where glioma cells grow in the context of human brain environment within the organoid called as GLICO, which is short for Glioblastoma cerebral organoid. Our findings were first published in Cell Reports in 2019, and we recently published a follow-up paper in Cancer Discovery. GLICO generation is a multi-step process which begins with deriving primary glioma stem cells from patient tumor biopsies, transducing them with a fluorescent reporter, and ultimately co-culturing them with mature cerebral organoids which were previously generated from HESCs or iPSCs. Within a couple of days after co-culture, these fluorescently tagged patient-derived GSCs start to infiltrate the organoid. Following invasion, these GSCs proliferate and migrate throughout the organoid, ultimately forming tumors that closely phenocopy patient's GBM. So co-culturing patient-derived glioma stem cells with organoids offer an innovative approach for microscopic observation of GBM tumor development in a dish. Here we show that GLICO supports proliferation of GBM tumors. 1 week after co-culture, GLICO sections reveal a hypercellular tumor bulk with an infiltrating edge of GSCs that are invading into the normal tissue, as you can see here. 2 weeks later, you can see that the organoid tissue is completely destroyed in GLICO, in contrast to the completely viable control organoids, recapitulating abundant necrosis observed in patient GBMs. Next, we pulsed GLICOs for 48 hours with EDU and performed IF staining to identify proliferating tumor cells. So these EDU and GFP positive cells show that glioma cells within the GLICO are actively proliferating. Moreover, GLICO tumors generated from 20 or 200,000 GSCs at 8 or 18 days post-co-culture show that GSCs proliferate within the organoids in a concentration as well as time-dependent manner. GLICO tumors also closely phenocopy human GBM's infiltrative behavior. So To model GBM invasion, we co-cultured 6 of our patient-derived GSC lines as 2D neurospheres or 3D GLICOs. So as you can see, for all the 6 lines, tumor take rate was 100%, but the growth patterns and invasiveness was unique for every patient in the GLICO model, which is depicted in this panel here. So 728 and 1206, the top 2 lines, are our most aggressive cell lines in vitro, and as you can see, they exhibited a highly diffuse pattern of invasion that extends to all areas of the cerebral organoids. In contrast, 320 and 810 GSCs, they actually formed quite large neurospheres in 2D and display an invasion pattern that is characterized by tightly woven honeycomb-like networks of tumor cells, which is quite distinct from the other lines I just showed. And finally, 517 and 607, as you can see, these have slower growth rates compared to the rest of the GSCs. And very similarly, their respective GLICO tumors were also characterized by smaller regional nodes with limited infiltration. So such strikingly different patterns and degrees of tumor cell invasion and proliferation between different GSCs and GLICOs reflect the phenotypic heterogeneity that is clinically observed in these patients. And moreover, this 2-photon image show that tumor cells that are marked with GFP penetrated pretty deep into the organoid, and you can see connected with other tumor cells. And as I mentioned before, in fact, in this Nature paper, which was published in 2015, it reported the existence of these multicellular networks in GBM tumors, where the tumor cells, which are labeled in green in this figure, are interconnected and supported by these long projections called as tumor microtubes. And these tumor microtubes were shown to facilitate tumor proliferation and invasion, and it also conferred chemo and radio resistance to GBM tumors. So we were able to actually recapitulate this biology in our GLICO model, where these long projections promoted invasion and proliferations of GSCs within the organoid. Next, to understand the ability of our GLICO model to recapitulate patient tumor heterogeneity, what we did was performed single-cell RNA sequencing of about 60,000 tumor cells from 5 different patients, and each primary GBM tumor was used to derive 4 different GBM models: glioma spheres, tumor organoids, GLICO, and patient-derived xenografts. So when we compared the model's transcriptional profiles to the patient's own tumor, we found that GSCs that were cultured in GLICO were significantly more correlated to the corresponding patient's bulk transcriptome as compared to the rest of the model. Strikingly, we also found that GLICO model has a higher proportion of cells in the proneural cluster, shown by blue in this pie chart, and a low hypoxic score highlighted by yellow as compared to the rest of the models, which is very encouraging. And moreover, we also found a significantly high stemness score in GLICO as compared to the other models. And lastly, GLICO was more similar to the primary GBM samples in maintaining the NPC-OPC subpopulation as compared to the rest of the models. And this is these findings are consistent with other published single-cell studies of the primary tumors. So this is really encouraging and shows that the GLICO model can capture the heterogeneity and molecular features found in primary tumors of patients. In addition to maintaining tumor heterogeneity, GLICO tumors also maintain key genetic features and molecular signaling networks of the parental tumors. Quantification of EGR copy number by DNA FISH revealed that 6 or 7 GLICOs maintain a high levels of EGR amplification that is seen in the parental tumor, but this amplification is actually lost when the same cells are cultured in a 2D setting. So showing that GLICO is able to retain this important molecular feature, which is very commonly seen in GBM tumors. Moreover, these phospho-RTK arrays of patient tumor tissue and corresponding GLICO for GSC lines 5 and 7 and 810 show that signaling profiles of the GLICO tumors are highly reflective of the signaling pattern found in the parental tumors. Next, we want to compare drug sensitivities in 2D and 3D cultures of GSCs against standard chemotherapy agents, which are frequently administered to brain tumor patients, such as temozolomide and carmestin, which is also called as BCNU. So dose response assays using 2D cultures of GSC lines revealed that most 8 to 7 and 9 to 3 GSCs have decreased cell viability in response to increasing doses of TMZ and BCNU. So this is for TMZ and this is the BCNU data. So we saw the decreasing viability in 2D, and next we wanted to see if we could see similar effects of TMZ and BCNU on GLICO tumors. So GLICO tumors were sensitive to BCNU and TMZ. However, the sensitivity, as you can see in this figure, is not as pronounced as it was observed in 2D culture. So, for example, 1 millimolar dose of TMZ reduced cell viability by approximately 90% in 2D culture, whereas it was only about 40% effective in a 3D assay. Similarly, 100 micromolar dose of BCNU reduced cell viability by over 90% in 2D, whereas it has absolutely no effect, as you can see here, in the GLICO. We saw similar responses for 8 to 7 GLICOs, which were treated with TMZ, except that 100 micromolar BCNU dose exhibited significantly reduced proliferation. But anyhow, this reduced sensitivity of our GLICO to chemo drugs is an encouraging finding, given that overall clinical response of chemo treatment is modest and chemo resistance invariably occurs during the course of treatment in these patients. We also compare the radiosensitivity of GSC lines grown in 2D versus in the GLICO model. So using clinically relevant doses of irradiation, we found that 5 and 10 grays significantly reduced cell viability as compared to the non-irradiated controls in 2D GSC cultures. However, if you see in GLICO tumors, irradiation with 5 gray did not really have any effect on tumor growth if you compare it to 0 gray. And 10 gray also could only slow tumor growth by about 25% or so as compared to the control, clearly demonstrating that GSC lines are more resistant to drug and radiation-induced genotoxic stress when grown within the microenvironment of the cerebral organoid than when it is grown under traditional 2D culture setting. So overall, GLICO is a valuable tool that can be used to precisely model the tumor biology and test potential drugs in a personalized setting. GLICO recapitulates the phenotypic and molecular heterogeneity of these parental tumors that I just showed you, which makes it a unique tool for studying GBM biology in the context of a natural tumor microenvironment, and can be used for exploring tumor host interactions, which other 3D models fail to capture. GLICO can also be used as a precision medicine tool for patient-specific tumor modeling and therapeutic intervention by pairing patient-specific tumor cells with their own mini brains as depicted here. This model is also easily genetically and molecularly manipulated and offers a robust platform for high-throughput drug screening of potential anti-glioma drugs. It also provides a valuable tool for novel target identification and validation, and can complement other preclinical models for understanding tumor and treatment-induced neurotoxicity. I also want to quickly talk about two additional approaches to generate GBM organoid models. Since brain tumors are characterized by a variety of DNA aberrations that cause oncogene overexpression and or loss of tumor suppressor gene function, the first approach is to generate genetically defined models of human GBM by CRISPR genome engineering. This approach basically allows recreation of the putative initiating genetic events and allows for the observation of natural history of tumor development in a cerebral organoid model, a process that obviously cannot be studied in human patients. And the second approach is to grow pieces of tumor as organoids, which I'll talk about in detail in a minute. So let's first talk about the genetically engineered organoids. Here, the authors used CRISPR-mediated homologous recombination strategy to simultaneously disrupt the p53 tumor suppressor gene and integrate oncogenic HRAS within the p53 locus. What they did was they co-injected p53 guide RNA plasmid along with HRAS expressing donor plasmid in 4-month-old mature organoid, and then observed the tumor initiation and development using a TD tomato fluorescent marker, as you can see in this cartoon. So the CRISPR-edited cells were 2 weeks after the electroporation, and they start to rapidly proliferate and show invasive phenotype from 4 weeks onwards, and kind of take over the entire organoid by about 13 weeks. The authors verified glioma stem cell identity of these putative tumor-invaded organoids by showing increased expression of OLIG2, GFAP, SOX2 markers, which are markers of GBM stem cells, as well as increase in proliferation by positive Ki67 staining in TD tomato positive electroporated cells. Finally, to confirm the oncogenic potential, the organoid tumor cells were orthotopically transplanted into immunodeficient mice, which exhibited disease pathology and uncontrolled invasive growth that led to the death of xenografted mice within 90 and 100 days, as shown in these plots. Next, in this paper, authors triggered tumorigenesis in human cerebral organoids by actually using two different genome editing techniques. Sleeping Beauty transposon system was used for oncogene insertion and amplification, and CRISPR-Cas9 system was used to introduce mutations or deletion of tumor suppressor genes. And the difference from the earlier study we just discussed is here the authors actually introduced the combination of plasmid into a developing cerebral organoid as opposed to a mature organoid before embedding them in matrigel. So what they did was electroporated the embryoid bodies at the end of the neural induction stage to exclusively deliver the plasmids to neural progenitor cells, or NPCs, which are actually believed to be the cells of origin for many brain tumor types. So they actually introduced a bunch of gene mutations. So they introduced actually 18 single gene mutations or amplifications, and 15 of the most clinically relevant gene combinations to induce tumorous overgrowth in cerebral organoids. So about a month after electroporation, out of all these various gene combinations, they observed striking overgrowth of GFP positive cells in 4 of the organoid groups, which either carried a MYC amplification or a CD cDKNA deletion and EGFR overexpression, or NF1, P10, and P53 deletion, or EGFR overexpression and cDKNA P10 deletion. So out of all those groups, these four had, a striking overgrowth of tumor cells. They further go on to show that these NeoCORs, or neoplastic cerebral organoids as they call it, recapitulated the cellular identities and invasive feature of GBM tumors. So in this figure, they confirmed glial cell predominance in organoid tumors by showing increased staining for GFAP and S100B, which are glial markers, as well as proliferative marker Ki67 as compared to the control organoids. And here they are showing the invasive edge at the interface between the tumor and normal cells that are marked with GFP and DAPI respectively. Finally, they also show that NeoCORs could potentially be used for targeted drug testing. So here they've treated the organoids with 5 different EGFR inhibitors and found that NeoCORs treated with afatinib and erlotinib showed significantly reduced firefly luciferase activity as compared to the DMSO treated NeoCORs. Finally, I want to talk about the tumor explant style organoid model, which is different than the cerebral organoid model, where either do through co-culture or for genetic manipulation you can trigger GBM formation. What authors did here was develop an explant style protocol to actually grow pieces of fresh tumor specimens as organoids without really dissociating them into single cells. So basically the resected tissue is cut into about 1 millimeter diameter pieces, and the red blood cells are removed, and then these tumor pieces are basically cultured in an optimized medium, which is without serum and matrigel on an orbital shaker to facilitate organoid formation, which takes about 1 to 2 weeks. And then tumor organoids can be propagated by cutting them into small pieces and then culturing them, so on. And they show that the organoids can be actually maintained for about a year by doing this repeated cutting and then growing of the organoid pieces. And then they name them as glioblastoma organoids, or GBOs, and show in the paper that GBOs retained histological features and cellular identities of their parental tumors, as you can see in the H&E staining and immunostaining for various cellular markers in basically 2 tumor GBO pairs in these figures. They also go on to show that GBOs maintained gene expression signatures of individual parental tumors in culture over time. So this shading here is showing you that the GBOs have been cultured for about 12 weeks, and they actually retain the gene expression signatures of all of these parental tumors over time. And here they're showing that exome sequencing analysis shows that GBOs can also maintain the mutational landscapes of their corresponding parental tumors, and single cell transcriptome analysis show that GBOs retained cell type heterogeneity of their parent tumors. Finally, they show that GBOs could be used for testing treatment responses in vitro. For example, treatment with EGFR inhibitor, gefitinib, for about a week caused reduction of Ki67 positive cells in GBS from 3 patients which had EGFR alteration. And here they also they also assessed response of CAR T immunotherapy in GBOs and show that specific targeting and killing by EGFR variant 3 specific CAR T cells in the co-cultured GBOs as compared to the control CAR T cells co-cultured GBOs. So Today we've reviewed various 3D GBM models with increasing complexity, from sphere-based models, tumor organoids, organotypic brain slice cultures, tumor explants, to finally using cerebral organoids to reconstitute tumor in a dish. So as we just discussed, GBM can be induced in cerebral organoids by co-culture with patient-derived GSC lines, or by genetic manipulation, or by culturing pieces of tumor biopsies on an orbital shaker. As you see, each model has something unique to offer in terms of understanding the glioma biology, and the choice of model system would really depend on the research question at hand. For example, GLICO can be used to predict the invasiveness, or in other words, lethality of primary patient-derived cell lines in organoids. Additionally, it can be used as a platform to study brain metastases in organoids by transplanting tumor cells from, let's say, breast cancer or lung cancers, which frequently metastasize to the brain. Most importantly, I think GLICO offers an unprecedented opportunity to create personalized solutions for GBM patients by matching patient-derived tumor cells with patient iPSC-derived organoids to create a personalized GBM model in a dish. Similarly, I think NeoCORs have some other advantages to offer in the sense that they provide a methodology for defined genetic manipulation of organoids to investigate key driver mutations and chronology of mutational events involved in gliomogenesis in a human context. It offers a possibility to test the role of tumor suppressors and oncogenes and analyze tumorigenic capacity of gain and loss of function mutations. However, it can also be a drawback, as specific genetic alterations do not occur in isolation in human gliomas, and therefore this model is not representative of the complex genetic landscape of human gliomas. Nevertheless, I think this model can be valuable to study effects of targeted therapy on tumors with specific driver mutations. In these two models, cells can be marked with fluorescent reporters to visualize tumor growth in the organoid in real time. And lastly, GBOs promise to preserve the local cytoarchitecture and native cell-to-cell interactions of original tumors and avoid clonal selection of specific cell populations in culture, since, as I mentioned before, they are grown in defined matrigel-free and serum-free conditions. And moreover, I also showed to you earlier that GLICO and GBO models have been shown to maintain original tumor gene expression and mutational profiles, and they also preserve the molecular heterogeneity and plasticity that is observed in human GBM, which is obviously fantastic to study specific questions. I think the biggest advantage of cerebral organoid-based GBM models is that they provide a neuroanatomically accurate human microenvironment to study pathophysiology of human tumors at a fraction of the cost, time, and resources when compared to mouse models. Organoids cannot replace animal models just as yet, but they can complement them in the preclinical phase to push the right drugs to the clinical pipeline. And like any model system, organoid-based GBM models also have some shortcomings, because cerebral organoids are currently limited by lack of vasculature, blood-brain barrier, and resident immune cells called microglia. But recent studies have shown that these cell types can be incorporated by co-culturing organoids with stem cell-derived endothelial and microglial cells to recapitulate neural, vascular, and neuroimmune interactions in the brain, which are very important for tumor pathogenesis. And additionally, tumor-on-chip models with microfluidics are also being developed to recapitulate tumor physiology and is allowing to expand the experimental possibilities with these systems. I want to close by saying that continuous interest and improvement in organoid technology is leading to a paradigm shift in studying GBM biology. For the first time, we have a robust human model of GBM that is allowing us to replicate the process of brain tumorigenesis ex vivo in a dish and predict patient responses to therapies to hopefully offer people suffering from this terrible cancer a new and personalized way to treat their tumors. With that optimistic note, I would like to end and thank you all for listening. Thanks.